Hi, my name is Pablo, and today I'm going to talk about the mucosal serotonin reuptake transporter expression in irritable bowel syndrome is modulated by gut microbiota by mast cell PGE2. The microbiota is the set of microorganisms that live in our body. The intestinal flora fulfills a lot of functions that we normally do not give importance to. It helps you with digestion and fermentation, producing energy by metabolizing carbohydrates. Also, it helps us to digest what we cannot. It synthesizes vitamins, for example, the vitamin B12 and vitamin K. The microbiota also forms amino acids and helps the absorption of iron and calcium. In the colon, it works as a defense barrier against infectious agents. The microbiota helps the immune system in the process of recognizing which bacteria it leaves alive and which ones it eliminates. The microbiota is the key so that you do not respond and attack your own cells, as happens in all autoimmune disease, where your own defenses, your own immune system is attacking you. This immune system must recognize each day if the millions of bacteria that enter the body are beneficial or harmful. If it spends most of the day recognizing bacteria that are harmful, it will exhaust itself and it will also prevent the transport of serotonin. This happens because a protein called TLR2 is going to affect the ability to transport serotonin, and this can change your mood for no apparent reason. Basically, it means that if you have a bad flora and I am constantly activating my immune system, this is going to affect my mood and my cognitive capacity. So now, what is irritable bowel syndrome? Irritable bowel syndrome is a problem that affects the large intestine. It can cause abdominal cramps, bloating, and changes in bowel habits. Some people with this disorder have constipations, others have diarrhea. Some people go from constipation to diarrhea. In the general objectives, they investigate whether mucosal SERT is modulated by gut microbiota via mast cell prostaglandin E2 pathway. So now I'm going to talk about the methods that they use in this experiment. The first one is they have to make a collection of mucosal and blood specimens and assays of supernatants. Colon samples from experimental animals were collected. Blood samples were collected by cardiac puncture and placed in heparin-coated tubes. The plasma was collected after the centrifugation. In Western blot analysis, we can say that Western blot is a laboratory technique used to detect a specific protein, enzyme, or hormone in a sample or blood or tissue, and the method involves the use of gel electrophoresis to separate the proteins in the sample. The separated proteins are transferred from the gel to the surface of a membrane, and the membrane is exposed to a specific antibody against the protein under study. Reverse transcriptase polymerase chain reaction studies, the PCR. So the PCR was designed as a test that helps me to identify a specific sequence of genetic material. In the specific case of RT, it is being assumed that a reverse transcription must be carried out. What we are evaluating is gene expression, because we start from an RNA that's coding for a protein. So what I have to do is return the information to find out what genetic code it has. Then we have the immunofluorescence staining. Immunofluorescence staining is the most frequently applied technique to detect and visualize various molecules in biological samples. When I see blue, I am painting with the API. When I am with green, I am painting with fluorescent. When I am with red, I am painting with rhodamine, and each antibody will have the color. The color will remain when the specific antigen for that antibody is, which is why it is painted in those colors. So here we have the results, and in the letter C we see a Western blot observed in distal colon from mice treated with HC, or IBS-DFS. The gut DH is the control center, and here we see how is a greater expression of the SERT protein in HC than in IBSD. In the letter D and E, we see an immunofluorescence staining for the SERT, and it was performed on section of normal mouse colon. SERT staining is in green contrasting with counter-staining nuclei. SERT is principally expressed on the surface area of the colon mucosa, but not in the lamina propria. In the letter F and G, we see an immunofluorescent staining to SERT, and E-cadherin was performed on section of normal mouse colon. SERT staining is in green and E-cadherin in red, contrasting with blue counter-stained nuclei. And in the letter K, we see the Western blot of TPH1 in distal colon from mice treated with HC or IBS-DFS. And what is TPH1? TPH1 is tryptophan hydroxylase, and this one is an enzyme that plays a role in serotonin biosynthesis. In this figure number 3, we have in the letter C another Western blot observed in distal colon from mice treated with intracolonic infusion of HCFS, IBSDFS, or PGE2. This is the same as the previous graph. The bigger and darker it means that there is greater expression. So in this picture, we have the authors that talk about the microbiota, and we can see if they are agree or disagree. As an example, we have that tag et al. They say that acute inhibition of SERT by the selective serotonin reuptake inhibitor increases colonic motility and contraction, which are associated with abdominal cramping. As you can see, this author is agree with the seminary title. And the same with the other authors. As you can see, they are all agree. So, as a conclusion of the benefits, pros, and cons of not taking proper care of our intestinal flora, there is a very specific type of bacteria that is crucial to properly protect your intestinal mucosa and not to nourish a good intestinal flora, which is a bacterium called Akkermansia moncinifera. Thanks to molecular biology, we realized that this bacterium lives in the mucus secreted by the cells of the intestinal mucosa. These cells produce mucus to protect your mucosa. Normally, this bacterium consumes this mucus as a food source. Contrary to what you imagine, this is not a bad thing. It is a good thing because with this consumption of mucus, the production of the intestinal mucus will increase. There is a very complex relationship between the bacterial flora and the intestinal mucosa, where the vast majority of your digestive system is there waiting for problems. The microbiota is very complex and defines what things I'm going to attack, what things I'm going to cause inflammation, and if this inflammation is key to cellular aging, if this process is out of balance, it will greatly accelerate the cellular aging process to a point where science is now studying a model of aging derived from a parasite called C. elegans. C. elegans has a 3-week life expectancy, so it is easy to see if there is something that is or isn't increasing that life expectancy. This parasite has a very simple digestive tract, and only when this intestinal mucosa that the parasite has is damaged do these 3 weeks of life expectancy begin to count. This means that if we take care of the intestinal mucosa in the parasite, we greatly extended the life expectancy of this parasite. And this is an example of how damage begins to occur in our body, and how cellular inflammation and dehydration begin to occur, also with the tissue oxidation, which are the 3 bases of any cellular aging process. So that's all for today.